Hello everyone, welcome back to Life or Fusion, where knowledge meets excellence. We are in the part 7 of Learn from PYQs for CSIR-Life Sciences lecture series. So let's start the question. So the first question is from part B. The question states that electron transfer from donor such as NADH and FADH to oxygen. Where the electron transfer occurs in the mitochondria in animal and chloroplast in plant. Okay, this is the electron transport chain. So where it occurs? In the transmembrane space of mitochondria and chloroplast, where you have different complex 1 till complex 4 and complex 5 is your ATP synthase. This is your ATP synthase. So this is a very simple question, but you have to use your mind very carefully. Don't be so hurry. So here they ask that where does the electron transfer occurs? So this that electron transfer occur in the intermembrane space of mitochondria and chloroplast. But the options are option A, membrane of ER, chloroplast and mitochondria, but not in the ER. So option A is the wrong statement. Option B and C both states that it's only in the chloroplast only, and here mitochondria only. It's not only in the mitochondria, it also in the chloroplast. So both B and C are wrong. So that's why option D is a common statement which says that organellar membrane means the organelle inside the cells which is having a membrane, okay, which possesses ATP synthase. Okay, it means that this option D is denotes to mitochondria and chloroplast, because mitochondria and chloroplast are the organelle inside the membrane, inside the cytoplasm. Okay, inside the cytoplasm, and they also have a membrane, and the electron transfer occurs in the membrane of mitochondria and chloroplast. So option D will be the correct statement. So read the questions very carefully while you are solving like this question. So second question. In both males and female, the gonads secrete polypeptide hormones called inhibin B, which inhibits. So in male and female, the inhibin B inhibits which hormone? This you have to remember this one. The answer is the follicle stimulating hormone. FSH is inhibited by gonads which release inhibin B. Okay, and inhibin B inhibits FSH where? In the pituitary. Okay, in the pituitary gland. Okay, is it clear? So one more simple question. The maximum frequency of recombination that occur between two loci means what is the maximum recombination frequency RF value is always 50%. So option B is the correct answer. Come to the now part C question. So it's an important question. So see the question. So expression of gene A is regulated by magnesium ion Mg2+. Okay, the expression of gene A in untreated N cells treated with Mg2+ was analyzed by northern hybridization and western blotting. A similar exercise was done for a mutant which was isolated with a 6 base pair deletion in the 5' UTR of the transcript of gene A. Okay, the following are summary of 4 possible results that are hypothesized to be obtained. Is it clear now? So the question states that the expression of gene A, okay, is regulated by Mg2+. Is it clear? So what is the role of Mg2+ in the gene and in the protein, because here they did northern hybridization for the gene, means for the mRNA, and western blot for the protein. So Mg2 plus binds to what? Your phosphate backbone in on the nucleotides. So what is do? It decrease the negative charge in the phosphate backbone. So during transcription, first let's see what is the role of Mg2 plus in transcription. Mg2 plus helps in transcription by assessing the RNA polymerase 1. Okay, it helps in the replication, fork formation. Okay, or your during transcription, it also helps in your for the formation of transcriptional fork. Okay, so that's why this Mg2 plus is very essential for transcription also. Then in translation, what is its role? Okay, in translation, it binds to the mRNA and helps in the formation of the circle, because mRNA made a circle and ribosomes binds to it and is released protein during translation. This is the translation process in the translation. So, and this Mg2 plus helps the binding of ribosome on the mRNA. Is it clear? So Mg2 plus is very essential for transcription and translation. And what is the role of Mg2 plus in 5' UTR? 5' on translated region. Okay, in the 5' on translated region of mRNA. Okay, in 5' to 3'. So in the 5' on translated region of the mRNA, the Mg2 plus neutralize the phosphate backbone of mRNA, so that it can help the formation of the circle by the mRNA, okay, and the binding of some ribosome in the 5' UTR. So if 5' UTR is not there and still Mg2 plus is there, then Mg2 plus will not binds to 5' UTR. Then no translation. Okay, is it clear? So let's solve the question. Okay. So what will happen? So if in an untreated cell, let's talk about untreated. So in a untreated cells, means in wild type and in mutant, what will happen? In wild type untreated means Mg2 plus is not there. It means that if Mg2 plus is not there, so what happened in untreated cell? So it can only give you the nucleotide. Okay, only you will get the mRNA. Okay, just mRNA you will get, no protein will be not there. So you see bands you are getting in the untreated cell in the northern blot in these two option. Okay, here also, but no protein. So this option is wrong. Here no protein, here no protein, and option C also now wrong. Okay, then so in treated cell you will get both the northern blood product, mean this mRNA, and protein. So both are present in the treated cell of wild type. Okay, not mutant. So now when there is a mutation, if there is mutation in the 5' UTR, 5' on translated region in the 5' UTR, then what happens? The RNA will not form this circle, so that this RNA will undergo degradation. Okay, so the RNA will degrade. So if RNA will degrade, means there no protein. So it doesn't apply to Mg2 plus, whether Mg2 plus is there or not, RNA will degrade and there won't be any protein in both in untreated and treated cells. So see here, there is no northern blot mRNA, no protein. So option B is the correct answer. So let's come to one very simple like this. Don't leave like this question. In part C, you will get easily 4 marks. A panel of 6 hybrid cell lines, each containing a different subset of human chromosomes, was examined for the presence of the gene product as shown below. Okay, so the cell lines are A, B, C, D, E, F, and the gene product presents are A, B. In A you have gene product, in B you have gene product, and in F you have gene product. So let's see. They ask that which chromosome is responsible for the gene product. So in A, you see gene 1, 2, 3, 4. All 4 genes or in all 4 chromosomes are there. Okay, and in gene or in cell line B, we have 3, 4, 5, 6, 7 still. Okay, when 1, 2, 3, 4 chromosomes are there, still the cells can able to produce the particular gene product. And when 3, 4, 5, 6, 7 are there, still the cells can produce the particular gene product. Means the responsible chromosome may be what? 3 and 4, which is present in both A and B. Next we'll check whether it's 3 or 4. So see in the cell line C, the chromosome 3 is present, but there is no gene product. And in chromosome F, in the cell line F, the chromosome 4 is present and there is a gene product. So which chromosome is responsible now? So chromosome 4 is responsible for the gene product. So answer is C. C, very simple. Next. Okay. So the body weight of rabbit is determined by pair of allele at two loci A and B. And they are additive in nature. So the presence of a allele have some addition of the particular character like height or weight, whatever. Okay, so these are called as additive nature. If more of this allele is there, then the effect will be more. If less of the allele is present, the effect will be less. Okay, so the rabbit with genotype A minus, A minus, B minus, B minus have average 1 kg body weight. So A minus, A minus, B minus, B minus have 1 kg body weight. Means when there is no allele, means no contributing allele. So this is called as contributing allele. That allele which will contribute to a particular phenotype like height or weight. So here the allele which is responsible for the will contribute for the weight of this rabbit is called as contributing allele. So A and B. So in first case, A minus, B minus, B minus were 1 kg means there is no contributing allele. No contributing allele. Okay, when there is no contributing allele, it's 1 kg. Then one more, A plus, B plus is 3.4 kg. Here, what are the things here? The thing is that here the contributing alleles are 4. So there are 4 contributing alleles here. How many? 4 contributing alleles means when all the alleles are present. Okay, when all the alleles are present, so the maximum body weight can be 3.4 kg and the minimum body weight of the rabbit can be 1 kg only. So this 3.4 kg happened because of the contribution of how many allele? 4 allele. 4 allele individually they contribute their contribution so that the rabbit became 3.4 kg. So how much they contribute now? We have to calculate. When nothing is there, 1 kg. When all alleles are there, 3.4 kg. So this all 4 alleles contribute 2.4 4 kg. Okay, so what is the contribution of each allele here? Because each allele will contribute equally. Equal effect will be there. So 4 allele means 2.4 divided by 4 equal to 0.6 kg. So remember, 0.6 kg was contributed by each allele. So without contribution, the rabbit will be 1 kg. With contribution of 4 allele, it becomes 3.4 kg. So each allele contribute only 0.6 kg. So what? So this means that if there is a single contributing allele, for example, if there is a single contributing allele, A plus, A minus, B minus, B minus. So means there is one contributing allele. So the weight of the rabbit will be 1 kg plus 1 kg the base weight or minimum weight plus 0.6 kg, which was given by the contributing allele. So it will be 1.6 kg. Okay, so the question is that a male rabbit with A minus, B minus, B minus is crossed with a female rabbit with A plus, B plus, B plus. What will be the predicted average weight of F1 progeny? So if you cross these two, what will be the F1 here? A plus, A minus, B plus, B minus. So what are the number of contributing allele? So there are only 2 contributing allele here. 2 contributing allele. So how much they will contribute? 0.6 into 2, that equal to 1.2 kg. So what will be the body weight of the rabbit without any contributing allele is 1 kg, with extra means 1.2 kg. So 1 plus 1.2 equal to 2.2 kg. So 2.2 kg is the correct answer. Next, in the enzyme linked antibody used in ELISA, the interaction between enzyme and antibody is stabilized by. So in the enzyme linked, what happened? You have already your primary antibody, they are binded already in the well. So now you have to add your enzyme. So enzymes will bind to the primary antibody. Is it clear? Then secondary will bind to the enzyme. So now this interaction, because you have to watch this, so this interaction should be very strong interaction. It should be a strong interaction. So which bond is the strongest strong bond? Is it hydrogen, ionic, covalent, or van der Waals? So all of you know covalent bond is the strongest bond. So the interaction between enzyme and the antibody will be covalent interaction. Next is a very simple question. Okay, your genetic mapping. E. coli cells were simultaneously infected with 2 strains of gamma phase. One gamma had a mutant host range, is temperature sensitive and known to produce clear plaques. That is HSTC. Another strain is gamma carried the wild type allele H plus, ST plus, C plus. Progeny phase were collected from the light cell and were platted on bacteria. The following number of different genotypes were obtained. So these are genotypes, these are number of plaques. Okay, so if you count, the total number of plaques will be 1000. So for genetic mapping, first you have to find what is the parental gene and what is the double recombinant gene. Okay, so as I told you in my previous videos also, the highest number of offspring or highest number of the F2 progeny which will show the particular genotype is the parental genotype. So this is parent 1 and this is parent 2 because 350 and 300. You can see here H, STC and H plus, ST plus, C plus are the parent, same thing. Okay, then what is the double recombinant? The lowest number of F2 progeny which will show the particular genotype. Their genotype is what? Double recombinant product. This is DC and this is DC. So what is the DC now? DC is H plus C plus ST. One more is H C ST plus. What is the parent? H plus C plus ST plus. One more parent, H C ST. I told you. Okay, so in the double recombinant product, two alleles will be from one parent, one more alleles will be from other parent. See here, H plus C plus from one, ST from other. Okay, H plus if I say parent 1, this is parent 2. So H plus C plus from parent 1, ST from parent 2. Similarly here, H and C from parent 2 and ST from parent 1. So what is the middle gene? So middle gene is the ST, and so the gene order will be H, ST, C or C, ST, H, whatever. So if you solve till now, still you can able to get the answer. See here, none of the option having ST in the middle gene. Option C. See, option C is the correct answer. But still we will solve this. So now, so the parent will arrange the parent as per the gene order. So the parent will be H plus, ST plus and C plus, then H, ST and C. So to find the recombination between or the distance between H and ST, you have to find the single recombinant product between H and ST. So what is the single recombinant product between H and ST? Okay, if you cross this, so H plus, ST and C, then H, ST plus and C plus. See here, H plus, ST and C. So this is the single crossover product between H and ST. Then H plus, ST and C. Okay, H plus, ST and C. Where it is there? Okay, this one, and one is H, H plus, ST and C, and H, ST and C plus. So this also your single crossover product between H and ST. Then this will be single crossover product between C and ST. This also single crossover product between C and ST. So what is the distance between H and ST? So total number of single crossover product between H plus and ST is, what is that? H plus and ST, 86 and 114. So 86 plus 114 plus total number of double crossover product, 4 plus 6. 4 plus 6 divided by total number of progeny, that is 1000 into 100. So what will be the answer here? 86 plus 114 plus 4 plus 6, you will get 0. So you get 21%. Okay, so 21% of recombination means 21 centimorgan between H and ST. Similarly, you find C and ST. C and ST is 90 plus 50. 90 plus 50 plus 4 plus 6 divided by 1000 into 100 will get 15% or 15 centimorgan. So see here, H to ST 21 and ST to C is 15. Is it clear to everyone? So hope you are liking our videos. Do subscribe and share with your friends so that everyone get help for a CSIR examination. Thank you everyone and have a good day.